You often think of mitochondria as simply floating around inside the cytosol, but in fact most mitochondria move in defined paths along the cell's microtubule network. Microtubule structure is remarkably dynamic and is constantly being remodeled. This remodeling of the network is important for the cell's ability to respond to metabolic changes or metabolic stress. Depending on these metabolic needs, mitochondria can be redistributed in the cell via this dynamic network. Mitochondria are also quasi-independent from the rest of the cell. Rather than relying on the smooth ER for lipid synthesis, for example, mitochondria can make their own lipids, which is a necessary process for the division of mitochondria. The spatial distribution of mitochondria within a cell can be quite distinctive from one cell type to another. In neurons, for example, there's a specialized area called the axon hillock, which is enriched in channels and transporters, all of which require ATP. Thus, there is also an increased concentration of mitochondria in that region of the neuron. Now that we have introduced the components of the mitochondrion, we will be able to appreciate just how intimately its overall structure is linked to its function as the powerhouse of the cell. As we will see, the double membrane structure of the mitochondrion is precisely what enables it to harness chemical energy in the form of ATP. Let's begin by looking at a very important series of chemical reactions that happen within the mitochondrial matrix, that innermost volume of the mitochondrion. As we will see in the next lesson, the food we eat is digested into simple sugar molecules called glucose. Following a meal or a snack, high concentrations of glucose produced by digestion float through our bloodstream until the glucose in the blood is absorbed by our cells. Cells begin to break down glucose to harness energy in the cytosol through a series of chemical reactions called glycolysis. The product of glycolysis is pyruvate, and the breakdown of a single glucose molecule into two pyruvate molecules generates a small amount of ATP. The pyruvate that was produced by glycolysis, on the other hand, is transported into the matrix of the mitochondrion, where it can be broken down more fully to generate many more ATP molecules than is produced by glycolysis.